Auzubillahiminashaitanirrajim. Bismillahirrahmanirrahim. This is Muhammad Tariq, and you are watching my YouTube channel MTK Journey. Today is our class 7th lecture on YouTube and class 11 biology. And today we will discuss about yesterday topic, technique used in cell biology, and inside the technique used in cell biology we will discuss today the second technique that we use in cell biology for observing the thing is centrifugation. Centrifugation means making parts. So it's also known as self-fractionation. Why this is known as centrifugation? Because it works on the principle of centrifugal force. And second one, it is known as self-fractionation. Self-fractionation means on this process we divide the cell into different parts. We separate from cell a different component. So this reason this is known as self-fractionation and centrifugation because in this process the instrument centrifuge work on the principle of centrifugal force. Definition is that a process through which we separate a different component of the cell by their shape, their structure, their density with the help of centrifuge machine. A process in which we separate a different parts from the cell, and also we separate a tissue from tissue. Different type of cell we separate with the help of centrifuge machine. So it work on the principle of density, on the principle of shape, and on the principle of structure. Okay, so what is the steps that we take to do centrifugation process? So very important, must be remembered. Step number 1, we will form formation of homogenate. First of all, we will form homogenate. Homogenate means we will form a mixture which is homogeneous, which is in homogeneous mixture, which is completely soluble. This like homogeneate mixture we will form first of all. By what? By mortar and pestle. By mortar and pestle we will form homogeneate mixture. Okay. In homogeneate mixture, we use a grind of tissue. When we do grinding of tissue, so in this process we use a liquid nitrogen, because we use a liquid nitrogen during grinding, during homogeneate mixture formation, because it make the solution, it make the tissue cool, and it also helps us to cool up. And also you can use isotonic solution, some type of isotonic things we can use during centrifugation process, because we use liquid nitrogen because it make cool. It's very it make in different laboratories method we use liquid nitrogen. That is very important, because up to minus 196 degrees centigrade it can stay in liquid form. Step number 2, we will put these homogeneate mixture into a centrifuge device. And the third, we will separate a large particle, and we will discuss and diagram later. And the third step, that we separate large particle inside a centrifuge machine. Number 4, separation of intermediate particle, and separation of small particle, and separation of very small particles. Okay, come to diagram. Let me to discuss from diagram. First of all, we will take tissue, and from tissue we will make homogeneate mixture. By what? By grinding process with the help of mortar and pestle, and with the help of liquid nitrogen. We will put the liquid nitrogen as well. Homogeneate formation. So we put the homogeneate formation in a special type of apparatus, and after this we will give 800 gravity speed up to 10 minutes. Up to 10 minutes, 800 degrees centigrade, 800 gravity speed, that will going like this, and up to 10 minutes. And after this we will put again this thing into another apparatus, and there will be a two type of thing will be separate from each other. Number one is supernant, and number second is pellet. Supernant is a thing that on the basis of density we first of all discussed, and the basis of density and the basis of structure and the basis of shape. So supernant go upward and pellet will go downward. In this process, first of all, we say that we will separate the large particle, and the large particle inside the tissue is maybe whole cell or nucleus. So on 800 gravity speed, up to 10 minutes, there will be two type of things separate. Number one is nucleus, and number two is a whole cell. And number second process, we will give again 20,000 gravity speed up to 15 minutes. And this process, after this, there will be two type of thing come. Number one is mitochondria and endoplasmic reticulum, and what? And pellet. Pellet, the downward solution is known as, the downward homogeneous mixture is known as pellet. And this pellet will be separate a different type of organs, organelle, cell, nucleus, different type of thing. And this is known as serpentine. Serpentine mean that will again we use another apparatus. Okay, so after this we will give one leg gravity speed, and one up to 1 horse, and there will be microsome. Microsome will be separate. Microsome mean it is a fragment of endoplasmic reticulum with ribosome. When we doing grinding process, during this grinding process, some fragment of endoplasmic reticulum attached with ribosome. This fragment which attached with ribosome, this fragment is known as microsome. So in one leg gravity speed up to 1 horse, there will be microsome separated. Microsome will come on pellet. After this, we will give another apparatus of 1 lakh 50,000 gravity speed up to 3 horse, and we will separate a very small organelle which is known as a ribosome. So at the last we will separate ribosome. In class 11 up to ribosome there is a centrifugation process. After ribosome separation, we can do another experiment also on this. We can separate another very small particle on this centrifugation process. Okay, so it work. First of all, I tell that it work on the principle of centrifugal force. Centrifugal force, a type of force that compel a body to, for example, there is a body that move in a circular form. So that force from outside, another force is act on that body to compel that body outside from axis of rotation. For example, this is a body that move like this, and another force that force act from outside and separate and help us, and that force act on this body to separate from axis of rotation. This is known as centrifugal force, and this is known as axis of rotation. So hope you will understand well, and in the next video we will discuss another process, chromatography, and another video. Inshallah, we will see you, and subscribe my channel and also like the video, and see you in another video. Bye.